Welcome to today's Seed World Innovation Series webinar. My name is Alex Martin, and I serve as an associate editor for Seed World, and today I'm happy to be your host. Today's theme is to have your breathing cycle with Computomics machine learning technology xSeedScore. I'd like to take a minute to thank Computomics for partnering with us on today's Innovation Series webinar. We have two lovely speakers joining us today for our webinar. Presenting today's webinar is Sebastian Schultheiss and Jörg Augmann. Our first speaker of the day is Dr. Sebastian J. Schultheiss. Sebastian is Managing Director of Computomics and founded Computomics together with a very experienced board of scientific advisors from ETH Zurich, Max Planck Institute for Developmental Biology, and the University of Tьbingen. Sebastian studied bioinformatics at University of Michigan and Tübingen. He worked on machine learning research and its application to biological data for his PhD degree at the Max Planck Institute for Developmental Biology and FML. He brings startup experience, bioinformatics skills, and machine learning expertise to Computomics, which brings superior prediction accuracy and unprecedented integration of phenotyping, genotyping, management, and environmental data together to agriculture, enabling its clients to produce stable, value-added crops. Our second speaker of today is Jörg Augmann. Dr. Jörg Augmann is a bioinformatics scientist at Computomics. He studied bioinformatics at the University of Tübingen and McGill, Montreal, and graduated with researching the evolution of epigenetic marks in plants at the Max Planck Institute for Developmental Biology. His broad experience in genomic data analysts and using machine learning techniques together with his training in plant biology fueled him to advance the technology on accurate phenotyping prediction and accelerate plant breeding for changing climates. During today's presentation, you'll likely have some questions for Sebastian and Jörg. Please type these into the chat box at any time during the webinar, and we will address them during the Q&A session we'll hold after they both finish their presentation. We'll also have some poll questions for you during the presentation today. These are going to pop up in your chat box window for you to answer. Today's webinar is being recorded and will be made available at seedworld.com following the proceedings. We will also be sending out an email with a link to the webinar, just in case you miss it on our website. xSeedScore predicts phenotypes, integrating diverse data at an unprecedented accuracy. Today, we're going to gain a better understanding of why machine learning outperforms standard statistical methods, how machine learning accelerates your breeding program, and how to exploit opportunities for different breeding programs. And with that, we're going to throw it to Sebastian, our first speaker of the day. So please take it away, Sebastian, and thanks for joining us.
Great, Alex. Thank you for the introduction. I'm Sebastian, the Managing Director and co-founder of Computomics, and I'm here today to talk to you about our company and about machine learning, of course, which you've already heard a bit about in our biographies. We've been researching machine learning for quite a while, and we are applying it to breeding. And that's what brings us to our product xSeedScore. And Jörg is going to talk us through a few examples of how we use hybrid and line breeding programs, and we support them with our machine learning technology. And I'll be closing up with how to work with Computomics, so you'll get an overview of possible collaboration opportunities. Computomics is guided by a mission of helping our clients to make data-driven decisions and accelerate the product development that they have in place in order for them to create products that are used in sustainable agriculture. We are supporters of the United Nations Global Compact, like many of our clients, and we also support the United Nations Sustainable Development Goals. Computomics was founded in 2012 by me and some colleagues from different research institutes like ETH Zurich and the Max Planck Society. And we are all experts in machine learning or plant research or bioinformatics. And the co-founders have contributed their technologies, for example, on interpretable insights into genomic data with the help of machine learning. And in over 130 projects with about 40 customers in 12 countries, we were able to showcase the successes of machine learning research, of applying it to real-world breeding programs, and of then seeing its success and the accuracy of the predictions in the field. Another aspect of why people choose to work with Computomics is that we guarantee data security. That means all of the data that you submit to us will always belong to you, and it's only stored on our secure hardware that we own, that's under our direct control, and that's located in a data center here in Germany. We focus on applying AI to genetics and phenotyping data, and also microbiome and environmental data sets. So some of the products that Computomics offers are listed here, and they're fueled by machine learning in different aspects. So xSeedScore, you will hear a lot about today in this webinar. We also offer a phenotyping solution called CropScore, and genomic annotation tools like AnnoScore and MethylScore that use machine learning in their own way, as well as Megan and Morpheus for metagenomics analysis, and in a few other projects as well. So all of that is based on different aspects of machine learning technology that we have developed and that is proprietary to Computomics. And this is really the important part. This is what sets us apart from other companies. And this is where we think it's an important aspect of today's webinar. That's why I'm going to spend some time on this slide to explain exactly why we know that machine learning matters and how we have proven that in the field. So this is proprietary technology developed by our co-founders and in-house, and it's under constant development. We keep improving on that all the time. And what these learning algorithms do is they consider all possible combinations. For example, in breeding between marker data and the environment to predict very complex traits. And that enables the following points to become true. They give you a completely different perspective on the multidimensional correlations between genotype and phenotype, and this improves over time. So on the one hand, we have very accurate predictions already early on. When we do parameter optimization, we run the algorithm again and again on different parameter settings, and each time we learn about the data structure, and we can improve our model so that through cross-validation we can show that it's really the best possible. We currently have these type of data that you can see here on observed phenotypes versus predicted phenotypes in different years. So from 2012 to 2018, we kept adding information over time from our breeder who has supplied these data from a breeding program. And what we are able to see here is that over time, the predicted versus the phenotype measured in the field gets more and more accurate. So by 2018, we were able to get to 76% correlation between our predictions and what is found in the field. And only by having this high accuracy, with these gains can be seen in this technology. The machine learning model is able to learn and improve over these several iterations and build a very general model for exactly this germplasm. So that's also why we never share any data between breeding programs, between customers. All of these models are proprietary to you. You own the intellectual property of them if you work with us. And what we share with you are the results of these predictions. And we never share any data that didn't come from you or integrate any data across several clients. Our goal is that this machine learning algorithm is learning from reality, whereas with statistical methods, they often just build a simplified, biased view of reality and take for granted certain distributions. And the machine learning method, in contrast, is able to even model very complex relationships between traits, even is robust against very noisy data, and therefore can also incorporate historical breeding records. And it's able to integrate heterogeneous information. And then the really great thing about our technology is that once we have this trained model, we can predict billions of samples of in different locations, for example, under different climate conditions within a few hours. So that's a very straightforward way of really going into very high numbers of predictions. And the other great advantage of our proprietary technology is that we can visualize phenotype contributions of data from different sources. For example, we can visualize different markers and what roles they play, and we can visualize effects of different climate settings on phenotypes. And that's higher order correlations. It's not just one-to-one relation between a marker and a phenotype. It's pairs of markers, triplets, 4, 5, 6 of them acting together on a phenotype, just like the biology suggests. And thank you for answering the poll question just now. It seems that all of you believe that machine learning is going to play a key role in the future of plant breeding, and we couldn't agree more. And even the big ones are starting to notice. In this case, we're looking at Google CEO, former CEO Eric Schmidt and George Church. They gave an interview last month about whether machine learning is going to be an important part of breeding in the future. And they said, yeah, it's about time that we start using this, because biological data is very messy, but new methods that they have discovered are helping them to make sense of the mess. And our answer is that we are using these methods successfully on biological data since 2012, and it's no longer a challenge for us. We have even validated these results in the field since a few years, and that's what really sets us apart from any other competitor. There's maybe just starting to use this technology, or they're still using statistical methods. So our approach is machine learning based, and it also has certain advantages, of course. Thanks to this high accuracy, we are able to put plant breeders in control and model the performance of markers and also of crosses in specific locations with this technology, delivering an accurate picture of the next commercial variety that you want to put into the market. And this really enables you to get a very high speed up in the breeding program. We make that possible by incorporating multiple data sources and thereby making these predictions very accurate. As I mentioned, we can accelerate breeding by predicting yield per location, for example, in actual numbers, not just a breeding value. We can predict accurate phenotypic values also in untested locations. So if you have results from other genetics that have been tested in a certain location but not in another, we can basically transform and virtually plant these lines in those untested locations and give you an estimate that is still very accurate. We can improve traits by identifying genes with the interpretable part of our machine learning technology, and that enables you to find maybe new gene editing targets that could be used to improve breeding even further. We've verified this in rice in our joint project with the International Rice Research Institute, IRRI, in the Philippines, in this partnership. And we also currently are serving 8 returning customers that are commercial breeders and that are using this technology already today. So they are already able to exploit the full genetic potential of their breeding program and decrease their testing by 50% or more. And with these twice as accurate yield predictions, they're able to really get to accuracy rating compared to the field measured phenotype of 76%. In some cases, we've even reached 80-85%. The very complex phenotypes are very difficult to predict for statistical methods, especially across different locations, for example, and this is where our method really stands apart. We have a very efficient method in place, so our breeding customers often don't just dismiss or deselect 50%, the bottom 50% of their breeding program, but in some cases even the bottom 99%, and only keep the top 1% of predicted lines, because thanks to our ability to model also specific combining ability, we have a lot of genetic diversity still in the breeding program if you do this, and you can really rely on these predictions to be very accurate. So that's also something, an example that Jörg is going to present in a few minutes. So with location-specific predictions, we can also help out breeders who are looking into vertical farming, indoor farming, where they have a lot of control over the environment. And that's something that we can then simulate and recommend crosses. So with that, I'm going to show you these different technology tiles or data sources that can be pulled into xSeedScore. There are different aspects of this where we can use, of course, genotyping and phenotyping data, but also historic records, weather data, climate, soil information, information about management techniques and inputs. All of that is, again, how our model learns from reality and has a very, very accurate picture afterwards of the plant phenotype and how it can be predicted. So with that, I'd like to hand over to Jörg to show examples from hybrid and line breeding.
Hello, everyone. I will walk you a bit through how a typical customer project works with us. So from input requirements to our final deliverables. I will set the focus on some challenges of breeders we work with, one from each of the major breeding strategies, hybrid and line breeding. So I'll explain how we help solving these challenges and present some results from which our customers greatly benefited. So in terms of input data, all we need is simply a file of phenotypes and a file of genotypes. So for phenotypic data, we can work with all typical text file formats. So I assume yours is in this cluster of icons here on the right side. If not, drop us a chat message. Preferred and most efficient is a format where there's only one phenotypic value per row, so-called a long table format, which you see at the top table, because this is exactly the input format for xSeedScore. But most commonly used is maybe the format Microsoft Excel we see for most breeders. And we can, of course, convert this into our needed machine-readable format for you. So in addition to the actual phenotypic values, our xSeedScore model can integrate all kinds of diverse metadata to each measurement, as Sebastian mentioned already. So like location and year, which is included in the table already, but also data that people maybe only start to collect now. So for example, climate data like temperature, precipitation, field data, or soil condition, and so on and so forth. So because including metadata offers more potential to explain a phenotype, and it allows for stratified predictions. So for example, location-specific predictions or climate-specific predictions. So in terms of genotypes, they can be as well flexibly provided and encoded. So we can work with a specification of alleles like single nucleotides, ACGT, or encoded as AB, for example, or with numeric values as the second table at the bottom. So we can, of course, work with heterozygous allele. You see the 0.5 value, and with imputed values. You see, for example, the last cell is 0.96. We, of course, will also impute missing data if needed. Typically, we use accurate expectation maximization algorithm. So genotypes can derive from all kinds of sequencing techniques. So from microarray chips to whole genome, whole exome, or targeted sequencing like RedSeq or genotyping by sequencing, GBS. So we have strong genomics expertise. So processing sequencing data and identifying reliable markers is one of our daily routines. So we're often asked how extensive the data should be to be suited for machine learning methods. A general answer is, of course, impossible because it strongly depends on many factors like species, trait, computational algorithm, genetic similarity between training and testing set, and how balanced the training set is, and so on. So, but as a very rough rule of thumb, our experience tells us that model accuracies show less variance from 1,000 individual genotypes on. So we see generally good performance with over 1,000 samples. In terms of markers, the model performance increases with the number of markers as well, but only to certain thresholds. And so often model accuracies gets lower again beyond 5,000 markers. So 5,000 is typically sufficient. A generic minimal number of markers depends on how polygenic or quantitative your traits of interest are, or how big the genome is, of course. And so we worked already successfully with 500 markers, but maybe with 1,000 markers, you're more on the safe side, because then we can also consider downsampling, so-called feature selection, and with that have also good chances to optimize model accuracies. So once we had a look at your data, did some quality checks, and assessed that this data is suitable to meet your goals, we can move on to the challenges you're facing with and how xSeedScore can help solving them. So I want to showcase 2 example applications that highlight the strengths of xSeedScore. So first I describe how xSeedScore accelerated a hybrid breeding program. So here we show a hybrid breeding scheme from one of our customers. So the goal was to combine 4 different genetically distantly related populations into one breeding population. So here you see the A plus B and E plus F populations. Those are the female heterotic pools, 2 heterotic pools, and CD and GH are the male heterotic pools. So you test your lines in this setting in each heterotic pool against individual tester lines from the other side to estimate their general combining ability, GCA, right? And then, based on the GCA values, you select new tester lines, and only after that, cross the other side again against those new testers in the following years. So, for example, here from all the crosses AB, so from all crosses of the AB females to the male testers 1 and 2, you select new female testers 3 and 4, only after field testing and double haploid generation, and only then you can advance on the male side again. So you basically incrementally advance each side in following years. And if you do that for a few iterations, it easily sums up to several years, as you can see. So in the end of this process, which is not shown here, you have optimized your pools, and then you typically identify kind of a handful of top performing lines in each pool and cross them to each other to produce the final hybrids, from which you then select candidates for commercial use. And so here is how we transformed this strategy together with the customer to significantly have sped up this process. And so we do that by exploiting the nowadays immense computational resources paired with powerful machine learning techniques we developed in-house, as Sebastian mentioned. So with these tools in your hand, you can not only simulate and predict in silico, so computationally, the crosses against individual testers. You can easily simulate and predict all possible crosses between the male and female side with xSeedScore. So test crosses are a quick approximation to determine the GCA of lines, and the choice of tester lines is important in hybrid breeding, of course, since they are basically half of the breeding equation, right? So with all versus all predictions, in principle, you get completely independent of testers. And you can get a better estimate of GCA values by not relying, because you're not relying on a single cross only, but because you can calculate GCA based on all possible crosses. And so you can discard the low performing lines earlier and not even test them in the field, which saves you, of course, time and phenotyping costs. So all this can be implemented in a single prediction, so in a single year, because you don't need to optimize the tester lines anymore. And so in this scheme of our customer, this saved half of the time of their established breeding pipeline. And it worked well. So the customer included 4 commonly used testers in the population from which we simulated all the crosses. And 3 of the testers were ranked first, second, third in the resulting GCA predictions with xSeedScore. The 4th one was among the best 15 predicted lines. So this confirmed, firstly, the accuracy of xSeedScore. At the same time, secondly, the superiority of 3 of the 4 testers. But also it showed the potential in finding new suitable testers with xSeedScore because of the 4th tester being only ranked on place 15. So in terms of runtime, training a model takes, dependent on the population size, hours to a few days. But once the model is generated, it can be used for predicting a single data point in basically milliseconds. So if you parallelize calculations, predictions of millions to billions of genotypes is now well feasible with xSeedScore. And statistical methods hit their limits earlier because calculating a kinship matrix of millions of lines is computationally much more challenging. Now, the same principle of all versus all predictions can now greatly also help in identifying the best final hybrids of the optimized males and females. So at the end of this depicted process here. So doing all pairwise crosses between all males and females in the field is of course infeasible for the breeder. And so one usually picks just a handful of top performing lines, crosses them, sows them out in the field. So these crosses are depicted here in this matrix as the dark gray cells. So each row and column in this matrix represent the optimized lines from each heterotic pool, ordered by the performance from top to bottom and from left to right. And each cell represents a hybrid derived from each parental pair. And so with xSeedScore, we simply now simulate all possible line combinations and predict their phenotypes. Here shown as all cells in blue of the matrix on the right. So let's assume the red hybrids in the left matrix are the top performing ones based on field tested GCA values, or if you wish, also the top predicted ones using statistical models. And the red hybrids in the right matrix are the top predicted ones with xSeedScore. So the selected hybrids in both matrices might well overlap, as is the case in this toy example here. But what you see is, first of all, some of the xSeedScore predicted ones are outside of the gray box, which are being field tested. So these could represent so-called hidden gems, so crosses between intermediately performing lines that have a high specific combining ability, right? And secondly, not all the gray hybrids in the left matrix are also red ones in the right matrix. So the prediction that some of the hybrids that usually get out into the field will not perform well can be of great importance as well, since you might decide to not plant them out and save the plot space completely, or use the space for more promising crosses. So to sum up this first part, so with xSeedScore, you can easily simulate and predict all versus all virtual hybrids of any population size. And importantly, you can do that in every step of your hybrid breeding pipeline. So you not only provide with that a better estimate of the GCA values, because you can average across the phenotypes of all crosses of a line, but you also capture cases of high specific combining ability, which can also be interesting targets to follow up on. And so, that is also the reason why machine learning can identify such beneficial combinations, because our machine learning methods account for non-additive effects. So meaning it learns the contribution of each individual marker plus a certain co-occurrence or any interaction between markers that is beneficial for a phenotype. And so as a consequence, xSeedScore can predict phenotypes of hybrids that outperform their parental phenotypes, which is not possible with the commonly used blood models, at least in their default formulas, where they capture only additive effects, and so they won't find hybrids that outperform their parents. So in our comparisons to standard GBLAB, we always observe comparable and in the vast number of cases superior model accuracy of the machine learning methods to a varying extent, sometimes more, sometimes less, but especially they outperformed statistical tools on non-additive traits like yield. Okay, coming back to our customer project. So the goal was to combine the male side with a genetically related female population. So one part of the scheme I showed you before. So we performed the in silico crossings of all of the 600 available males here versus all of 150 available females, which then multiplies to approximately 100,000 hybrids. So the training dataset consisted of the phenotypes and genotypes of the parents, but also of some hybrid crosses. And here you can see the matrix of before in a real case of a subset of the parents. So in red, the red cells are high performing hybrids, the blue cells lower performant hybrids. And so from those predictions, you can now accurately estimate the GCA of the parents, which is basically averaging over rows or columns. But you can also clearly, at first glance, distinguish well-performing from poor-performing parents. We can even cluster parents together that perform similarly well throughout the population, and therefore avoid choosing kind of redundant hybrids, because we can choose to only choose a few lines from each cluster, right? So in total, you can inspect the whole population and use it as a basis which lines can go with which other lines. And so this prediction worked really well, as I will illustrate here. So on this plot, you see the measured phenotypic values of two related traits on the X and Y axis. So the best performing lines would show up in the upper right corner. So the turquoise dots represent all predicted hybrids, and the dark dots represent the hybrids that contain around 10 top predicted males with xSeedScore. And so you see that those are strongly biased towards the upper right corner. So the customer, so from the lines that the customer selected for second cycle in his breeding program, 80% of the top male versus top female crosses, as predicted by xSeedScore, have been selected. And their performance strongly exceeded that of commercial checks by nearly 10%. So by contrast, the predicted medium-low crosses achieved only 92% of commercial checks. And so they have not been advanced to the next generation. So xSeedScore identified the top male lines that apparently combine very well with this new genetic material from the female side. And so alone from as few as around 10 top performing male lines, the customer could follow up on a few hundred new opportunities to find one commercial hybrid versus the usual 20 or 30 you get commonly, as he claimed. So that's because, if you recall, we now have predictions not only from the roughly top 10 versus top 10 field tested lines, so maximally just 100 hybrids. We can now evaluate all possible crosses of a top performing parent. So one could decide to take, for example, the best 20, best 30, or best 50 hybrids from this parent further into the next testing phase, and not just maximally 10 per parent. So the customer was enthusiastic about these results, and they reported to us a significant boost in their breeding program. And let me stress that again, in just a single year. So this was an example where we, in close collaboration, contributed to pushing a breeding program forward, and we're happy to learn about other programs and discuss how xSeedScore can fit in and help as well. So I'd like to switch now to an application of xSeedScore in line breeding. So the goal of line breeding is, of course, as you know, the homozygous elite line in the end. So you're not interested in hybrid genotypes, but if you cross lines to advance your population, you have to inbreed or backcross several times to make the line homozygous again. And in each step, you select for stack up beneficial traits. So this is again a case where a lot of simulated genotypes can help in selecting the right parents that will produce high performing offspring to a higher fraction or to a higher probability. So here computational power is again needed to simulate the actual F2s, F3s, F4s, in general, any FN generation. So here's a concrete example of a customer. So in a customer project, we simulated all possible crosses between 50 parents. So let's do a bit of math. So we have 50 times 50 divided by 2. So a bit over 1,000 F1s, or to be correct, 50 times 49, but these gives better, nicer numbers. And now if you inbreed further the F1s and generate 1,000 F2s for each individual F1 genotype, and then simulate every time 10 of the following generations, you quickly end up with over 100 million F4s, like in this example. This sounds a lot, but however, as Sebastian already mentioned, predicting these 125 million genotypes with xSeedScore is just a matter of minutes. So this is no challenge anymore. We can even then predict phenotypes for many locations. So for example, if we have 10 locations, the number of predictions multiplies by 10, and we still can feasibly predict all over 1 billion genotypes in just a few hours. So with location-specific predictions, as we already also mentioned, so xSeedScore is ready to explore questions like which parents will produce well-performing offspring in an extreme location if you have some training data of that location. Or if you have weather data, how will your plant perform in a 2 degrees increased temperature environment, for example? So the underlying principle of this simulation is the more genotypes you model, the more likely you find the well-performing combinations of markers in some parental crosses. And while this again helps in estimating the GCA of parents and basically predicting the performance of your breeding population based on those parents, this also paves the way to find an ideal genotype, basically, that is predicted to perform best of all the crosses. So maybe in some near future, one could fingerprint real crosses for similar alleles, and in this way kind of agglomerate similar haplotypes, which have higher chances to be high performing. So this would then be target mating. And this could even be facilitated or accelerated by genome editing in the future. So this just as a little outlook. So coming back to the customer project. So the customer was interested or used this basically like breeding simulator on their population and was interested in the prediction of 2 traits at the same time. So we predicted yield and maturity independently, and then plotted the predictions of maturity here on the x-axis versus yield on the y-axis in this scatter plot here. So now you see a lot of dot clouds. So the dot clouds basically with the same color are all the virtual offsprings from a single parental cross. And so how you can read this plot is that you will find the high yielding and early maturing genotypes towards the upper left corner here encircled in this plot. So those are the most promising for commercial use because they are obviously the most profitable. You can now basically, with this kind of plot, simply pick your preferred crosses, while you maybe want to retain, of course, a bit of variance in the trait. So not only pick from the top left corner, but you also nicely see that many clouds, so many crosses, never get near this beneficial region. So they might be readily discarded very early in your program. So for example, the low yielding ones at the bottom. And as a last side note, you can even interpret the different slopes of the clouds. So they indicate that for some crosses, maturity and yield correlate better than for others. So which can aid in selecting high yielding offsprings that are more independent of maturity than others, which was a central question for this customer. So for this project, we await the field measurements of the top predicted lines in the next year to assess how well this prediction performed. So these were 2 examples where xSeedScore helped in making informed decisions in selecting parents, testers, or in increasing the number of candidates for commercial use. So in each project with us, after we worked on your challenges and aligned with you all what we worked on in regular meetings, we will write up everything we did in a comprehensive report. So all our analyses are included, all data processing, quality filtering, what we did, and explanations about our methods, of course. So we have a routine set of helpful visualizations, some of which I presented, but we can also generate figures tailored to your specific needs or questions. And of course, you get all our simulations and predictions to download from our secure sharing platform. Because we're not a one-click solution provider, if you explain to us your concrete breeding program and challenges you have, we will strive in making sure you can directly use our analyses downstream without post-processing or touching the data again on your side. And so with that, I pass on to Sebastian again, who will follow up on how to work with us.
Thank you, Jörg, for this overview. So what we are going to talk to you about now is, I've also seen this come up in the questions already. How do we actually work together? So what's the goal for us when we start working together, and how can you actually access this technology? So when we work with you, we look at the experimental design, at the study design of the breeding program, and make sure that we can use all of the existing data, historical records, and of the samples that you're already taking every season. And we then discuss maybe adjustments to that if there is anything like this necessary, or we simply work with existing setups and use the machine learning and model training on your specific breeding program. And again, this is always going to be custom, and we never share this information with anyone else or use the information across different customers. What we then provide to you, as we've just seen, there's a variety of output formats. And to answer one of the questions is, yes, we can actually give you access to this technology. And there are a few different ways of us making that happen based on the different platforms that people might already be using. So we want to be your partner to fully use this these opportunities that AI brings to breeding programs, and we want to support you on this journey to use this efficiently. The first step would then be looking at genotypes and phenotypes, of course, but then also integrating location information, weather data, climate, or even causal loci if you already know. Here's a chart that might be more interesting later on when you maybe take another look at this presentation. This is how we can walk you through this journey. I think the most important part here is, again, we have a unique technology that allows you to get ahead in using very accurate predictions and bring big impacts on all of these different steps within the breeding programs by giving you more confidence in the selections that you can make in silico, and make better use of all of the field trial space that you have by using it exactly on those lines that have the biggest promising yield and the highest promising phenotypes in different combinations. So there's also, I guess, the question about genetic diversity. So what we can do with you by looking at the study design is define a goal function, let's say. So we can help you with identifying the exact goals that you would have in terms of minimal genetic diversity, for example, and then incorporate that into these predictions and make sure that we show you exactly how you're selecting. So in total, we currently have 17 different technology tiles developed, and we are constantly adding more of those. So there is always a possibility to work together on also helping develop this. The data security, again, is very important to us. So for large customers, we have the option of creating a joint venture company where there will be an on-premise installation of this technology. We can license it to you, and we then have access to, for example, royalty fees via this entity. The typical method today to work with us is on a software as a service basis. The data is actually being processed by us on our servers and delivered to you. So that can be integrated through an API into your own system. But the goal is really to have the predictions there as integrated as possible into your existing system, so there is no need for any adjustments in your workflow for the most part, and you can simply use whatever you have been using in the past. So the third option that we've outlined here is, if you really don't want to share your data with us, we have a serverless computing solution for that, where that's not necessary. We just both have to trust a third-party cloud provider, and then we can use this way of working together. So if you want to engage with us, we can start with a proof of concept to show you the advantages of xSeedScore in your data and specifically for your breeding program. You can then license this software tool, or we can work on a partnership model where we also cover all of the predictions and analyses needs that you have in addition to these to the specific xSeedScore technology, or we can engage in a risk-sharing model where we provide these predictions at a discounted rate and the licensing at a discounted rate in exchange for royalties that are then to be paid on successfully commercialized varieties that come out of the breeding programs that we help support. So the next steps for such a proof of concept would be we'll schedule a call to answer all of your questions, and anyone participating in this webinar will have a free consultation specifically for their breeding program that we offer, and if necessary, we'll form a confidentiality agreement so you can tell us about the data sets that you have, and we're happy to then answer all of your questions about it. We then provide you with a quotation. During the project, there will be regular exchange with your designated project manager and an analyst. So there will be, for example, weekly phone calls. A typical duration for a pilot project is just 1 to 4 weeks, and we then present the results and discuss any next steps. So with this, we'd like to end the presentation of the webinar, and we're happy to take any questions. We'll be in ASTA next week in Chicago, so there's still some open slots for our meeting. We'll be there in force, so there are several people from Computomics there that you can meet, including myself, Sebastian, and also Ruth and Anna will be there. They're happy to walk you through this presentation again or answer specific questions to your breeding programs. Thank you.
Thank you so much, you two. I'd like now to do a short Q&A session. So if you have a question for Sebastian or Jörg that you haven't typed in yet, please go ahead and type it into the chat box now. We have quite a few questions to get through today, so I'm going to work our best to get us through all of them. So I'm going to go ahead and start right off. Now, Sebastian, I think this is a question for you. It came through during your slides. How does your company solve the genome complexity of polyploid crops, especially regarding transcript and, excuse my pronunciation from this might be wrong, methylation?
That's right, yeah. So that's a very good question. We are working with plants for a very long time, and we have experience in over 40 plant species across a history of our company, many of them polyploid. So we do have a very good track record of dealing with these. We have proprietary solutions for many of these questions, and we have also specifically developed any tools for dealing with polyploid crosses and so crossing in polyploid species, and with transcript data and DNA methylation data in polyploids. We've specifically developed our MethylScore software that's able to capture the differentially methylated regions inside these between different samples, and that's what the software was specifically created for.
Perfect. Thank you. I think this next question also came through during your section, Sebastian, but Jörg, if you have an answer to it as well, feel free to jump in. Did you do any, and I'm going to make this question a little bit more broader because I'm not sure which slide this was mentioned on, but do you do functional analysis of genes predicted by the machine learning?
Yeah, so we have specific tools for functional analysis in place, unfortunately, many of our breeding customers are really only interested in increasing yield. So that's the main goal that many of these projects have, and that's what we then provide. I think an important part for looking at the different function is to see whether the genotyping data that we have is actually whole genome data or whether it's a representation of a part of the genome from which it was captured. So is there simply a marker that in the vicinity of possibly the causal modification or variant or allele of the gene, or is it the actual causal SNP that we were able to identify? And the second aspect of this is when we look at these whole genome data sets or even at representations, there are several methods that we have in place, and this is one of the projects that or products that we've offered for the longest time throughout the history of our company to look at causal genes for specific phenotypes. And there are many different ways that we have used and employed in the past to really get to that functional analysis. And often it's not simply a gene. It might also be a transcription factor binding site or change in a non-coding RNA. There are many different causes and effects that we have uncovered in the past for different clients, also for many different phenotypes.
Perfect. Thank you so much. Now, I think you actually addressed this question, but I don't see any harm in addressing it again. So is xSeedScore a platform that clients can access, or is it just run inside Computomics labs?
So for the most clients, running it on our servers is the most convenient, because while we do have very efficient algorithms, as we've shown, we can run many of these predictions within an hour or within a few hours, and therefore the turnaround time can be very fast. It's still high performance computing that you need, and that's usually not something that many clients will want to buy simply for the duration of a few predictions every year. So by spreading the workload of our high performance compute cluster across different clients, that makes it feasible for us to invest in very high quality computers. When we predict these results and send them back to you, you have, as you've seen, a lot of guarantees about the data security, so there is no reason to worry about that. But for large clients and for very sophisticated analyses, we are happy to discuss integration into the software system that our clients are already using, or simply licensing the technology, or integrating it via one of these serverless computing solutions so that it runs in the cloud.
Great, thank you so much. Another audience member asked, how do you leverage predicting the SCA candidates? And he added a note and he said he asks because farmers pay for SCA and not GCA.
So specific combining ability, as we understand it, is really the combination of basically higher order correlations between markers or alleles of different genes, and that's what makes it so difficult to capture for linear statistical models. And our model is able to capture that, and that's exactly what we often get asked about. Is this something that you can do? Is this something that you're able to provide to your clients? And the answer is absolutely yes. So this is something that we are happy to do, and that we are able to, as indicated in this question, this is often what farmers actually pay for. This is what they would like to see in the field, and these are the hidden gems and the unique combinations that we can uncover.
Great, thank you so much again. Our next question asks, does xSeedScore balance recommended predictions with genetic diversity?
Yeah, and I've briefly touched on that. So we do have different optimization functions, different goals for breeders, and we discuss the actual study design and the optimization that we report in our graphs with our clients before we start with the analysis. And that is something that we can integrate. There can be a minimum genetic diversity that's still maintained, or we can create a balance. So there are many different ways of doing that.
Perfect. Thank you so much. And then how do you account for the genetics of the crop? For example, genome rearrangements in crops such as canola.
Yeah, so this is, I think this has to be handled on the side of the marker discovery. So if you have a lot of duplications or heterologous regions, we would make sure that the markers are not encoded as 0, 1, but that you have a kind of a dosage. We used xSeedScore already on quantitative data, like on methylation data actually, which have between 0 and 100 all the possible values. And that would be then also the case. So if we can genotype the canola lines correctly, if we find the gene dosage, we can use this gene dosage for prediction, actually. So this I would address before applying xSeedScore.
Awesome. Thank you so much. Now, is there a minimum of data history needed to run computomics analysis?
No, absolutely not. We also do quite often year-specific predictions. So we just collect the data from one single year and let the predictions run on that. As long as your training or basically the whole population is large enough, sufficient enough, as we mentioned, fulfills the requirements, we can work on a single year as well.
Okay, perfect. And now this next question, I'm not quite sure what it's talking about, but I'm going to give it an idea. It says, how does it assist with disease resistance? And I assume that means xSeedScore.
Right. So what we mentioned in the final slides is we are able to pull in data sources from different pieces of information that can be combined within xSeedScore. And the great thing about this is that it can really be tailored towards your specific goal or the specific breeding goals and optimization function. So if you tell us that this marker or this disease resistance absolutely needs to be there, then that is something that we can model. If you have information about a beneficial change in the genome that is associated with a certain haplotype, that's something that we can put in there as well. If you are looking to discover new disease resistance, that's also something that the interpretable part of the machine learning is able to capture. So if we just briefly look at this backup slide that we created, here you can really see that for most relevant selection criteria, you can often see certain markers that are being picked out by the feature importance measurements that we do. This can be also combinations of markers. We can do this for higher order correlation. So this actually allows us then to detect disease resistance de novo.
Perfect. Thank you so much to both of you. I think that's about all the time we have today, but I'd like to take a minute to thank our speakers, Sebastian and Jörg, for joining us today, and for Computomics for making this webinar possible. And thank you so much to everyone who participated today in our webinar and for all of your wonderful questions. And we hope that you find this information of value. Again, a recording of this webinar will be made available later in the next couple of days at seedworld.com, and you'll also receive an email with the link to the webinar. Thank you again, and we just hope you have a fantastic day. This is Alex Martin of SeedWorld signing off.